We should start on time.
Let's introduce our next speaker, WendellGovea from Tufts.
Hello. So I'm a biologist, just in case anyone was confused. Woo! And I was trying not to talk about models, so I tried to put sense-making in my title. But then I realized I kind of probably needed to talk about models a little bit. So I decided I would have a new title, and this is my title. Because I think what's important to understand about Wendell, and I think everyone's going to make this point, is that while Wendell was deeply committed to the power and utility of models in classrooms, he also just really didn't care that much about models at the end of the day. What he cared about was, like David said, getting students to think, and he used this framework of models as a mechanism to get students to do that. And so when I met Wendell, just to give you some context, I was a graduate student in biology. I wasn't in education as I am now. And I started talking to him, and I went and visited what was then Physics 7 or class. And I walked into those discussion lab classrooms, and I was just blown away. Just literally, it had never occurred to me that a learning environment could look this way. And it really changed probably the trajectory of my entire life, walking into those classrooms. Because I walked in there, and students were talking and arguing and drawing on the board and making sense of stuff. And even just by being in that room, I started to understand physics better, just like by standing in there. And I was desperate as a biologist and a biology educator to get something like that going in biology, which, if any of you have ever taken biology, you may not remember a lot of thinking as part of your experience. Maybe you do, I don't know. But in my experience, not so much. And I really grabbed onto this idea of, it must be that these models are the key to me transforming biology. And if I can find those models, then I can change biology. And that's where I started way back in graduate school in 2007. And as probably many of us will say, Wendell, while he started off calling this a models approach, gradually and others kind of gradually moved models out of the center for reasons that I think I will now maybe make evident. But first I want to just give you a sense of kind of what it was like in those classrooms as best I can. And I was looking on my computer, and I found just a transcript from a class from Physics 7 from I don't even know when, probably 2010 or something. We were writing a NARS proposal. And this transcript is just like an average day in Physics 7. So you'd have a problem like this, which is, I don't know, probably standard in physics. I don't know what you guys do. But there's these rails, and one's wider and one's closer together. And the problem was for students to make sense of this. And if you look at what's going on in this discussion lab, here you see two women talking about physics. One of them has this intuition that maybe because one of the balls is sitting on a narrower track, it's a little bit higher up, and so maybe that's going to make it go faster because something about potential energy. And the TA comes in and corrects her and said that doesn't matter. But they keep going with their ideas. Another student is thinking about whether or not friction might matter. When they finally run the balls down the tracks, the student Andrea is right in her prediction that the thinner track will have a faster ball moving down, but she also understands that she was right for the wrong reasons. She doesn't yet know why, and they keep talking, and these are just snippets of this really long transcript of them continuing to grapple with this problem. This student, Bonnie, then brings up this idea of something about the spinning that they're noticing might be involved here, and Andrea is trying to explain her intuition that there's more movement in the ball, more of the ball is moving on the track in the thinner track. And they haven't learned about anything about rotational energy. I don't even know if that's a thing. But anyway, the point is there's all these ideas that are coming together under this larger energy interaction model framework. And it's not as if Wendell wanted students to just take a model and apply it to a problem. This is not just model application, right? He wanted them to synthesize some ideas from this general framework with their own intuitions, with their own observations, with what was going on, talk about it with each other, and that's the sense-making that Wendell was talking about. And that's the stuff that just blew my mind when I saw that students were doing this for 5 hours a week, okay? And then they just had 80 minutes of lecture. So this was the bulk of what was going on in this class. And then I got distracted by this question. And it's really hard to get distracted. It's really easy to get distracted by this question because it's such a weird, confusing term, what a model is, and it gets used in all these weird ways. And I just got obsessed, and I started reading the philosophy of science, which is never a good idea. And then I started looking around for the ways in which the word was being used in biology, and I got annoyed, which is like so unlike me. There I am in 2007. I was so annoyed. And why was I annoyed? Because I kept seeing this way of describing what models are. Models are abstractions. You still see this. Abstractions or simplified representations of real systems. And I was like, I guess, but that's not really what a model is to me. I was like, where's the ideas? Why are you talking about representing a system? Like, to me, models are all those cool ideas the students had about what's going on in the system. So I was mad about this, and I was mad about it, and just to illustrate kind of where I was coming from, this is a popular way of talking about a model in biology, the model of DNA. And look how pleased they are of themselves for having built this tin kind of structure of the molecule of DNA. And just so you're clear, I don't think this is a model. Okay, this is a representation of a molecule made out of tin. The model is the ideas about the mechanism that are embedded in this representation. And in fact, this is not the best way to represent those ideas. This is a distracting way to represent those ideas because you got all kinds of tin and other things flying all over the place, and you don't need any of that. What you need is, this is a drawing that Crick sent to his son in a letter. Like, here's what we figured out, basically. Like, there are these base pairs, and they pair with each other, and that means a molecule can hold information, and it can replicate information across generations. Like, whoa, that was the idea. And you don't need this fancy thing to think with that idea. And yet, what do we do in classrooms? We do this. And if you're really crazy, this. Right? And you have students build DNA models. And that was all there was in the biology curriculum in terms of modeling that I could find. And it drove me crazy. So the question that I kept asking is, like, what are you doing with these? What's the idea? What are these models for? And that led to this paper that I wrote with Cindy, because you'd think, oh, maybe you're just going into like really bad biology classrooms. That's why you're encountering all this like gumdrop modeling. But no, if you go and look at the next generation science standards and you look at the modeling, go ahead and do this. You just look at the modeling standards and you look at the way that they're written, and you will find the following things. These are verbatim quotes. Develop and use a model to describe the function of a cell as a whole and something, I don't know, parts. Basically, you can interpret it pretty easily if you're an instructor as just have them like build a cell with some parts in it. Or this, like develop a model to describe atomic composition. Just like build the ball and stick thing. Things that you're maybe already doing, and now that becomes modeling, and now you've checked off your NGSS standard, and you're done. But what are the ideas here? Like, why are you building a cell? Like, what are you going to do with that? What are you going to explain? So we tried to kind of hack this by saying, well, what are these models for? Like, what ideas maybe are in this space that could explain some phenomena in the world? And this was kind of a reach for some of these. Like, I don't know, maybe you want to be able to explain, like, what happens when cells malfunction or some other interesting phenomena about cells, or maybe you want to explain, like, various properties of substances or other things. But this is what we were trying to get at with this paper, models of versus models for. We wanted to kind of bring the ideas back into models and help teachers think about models as sets of ideas. And it took me 10 years to write this paper, just if I'm being honest, from that moment of being annoyed in 2007 to 2017 when this paper came out. And it would have been a lot faster if I just read like the intro to Wendell's textbook, because he just wrote this all out. Like and now I'm just going to put the citation here so you can just cite it, because he has a beautifully clear description of what models are just right in the beginning of his textbook. And he says these things right in the margins and these little bold cutouts, like, no, models are not descriptions of things in the world. That's not what they are. Equations and representations, those are not models. Okay, those are ways you can represent the ideas. That is what models are, right? And he had his big push was, models are these really powerful sets of ideas that students can think with. I know I can maybe stop belaboring that point now. But the reason he organized his entire course around models, or one of the reasons at least, I was thinking about it, and I know that he was a big fan of David Ausubel's work. And this Ausubel had this idea of advanced organizers, which is a way to think about how you organize your curriculum. And instead of organizing it however you guys organize physics textbooks normally, the idea was, why don't you present information in a way that's going to be sensible and usable to students? And really the way Wendell, his sort of brilliance in interpreting this idea of an advanced organizer was to say, there's only really a couple sets of big ideas that students need, so I'm going to organize the whole curriculum around these really powerful clusters of ideas. And then I'm going to draw students' attention to those cool ideas and say, if you have ideas that are related to these big sets of ideas, you can use this whole cluster of ideas to make sense of the world. And that's how we transformed, really, the curriculum. I spent a lot of time trying to do something similar in biology, and for various reasons, that has been very difficult, and I'm happy to talk to you about it if you want to talk about it. But really what I want to say is that Wendell's approach to modeling and model-based reasoning was really informal and really pragmatic, and I really loved this about him, and it made this whole approach very flexible and very adaptable, because models then became whatever tools, Wendell would say this all the time, whatever you need, whatever tools, whatever ideas, whatever representations you need to make sense of something, that's what the model is. And the real reasoning happens when you're actually trying to take all those ideas and apply it to some specific phenomenon that is confusing or puzzling or weird in some way. And that's where the work happens. So, finally, when I was confronted at Tufts with designing my own introductory lab curriculum, I started thinking about what I was going to do. So I now had a context where I had students who were used to doing experimentation, and like, again, if you've ever taken a biology lab, not really idea heavy, kind of more measurement of things, skill building, there's a lot of pipetting. And I wanted a way to bring ideas into these labs. And I was thinking to myself, how can I do that? Like, how can I get models into these labs? Because there aren't really like these just 10 canonical models that I can draw on. So I really struggled with that for a while. What I ended up doing was just letting go of this obsession with models, quite frankly, and started focusing on how can I just get some interesting phenomena into the lab with just any ideas that students have that they might be able to think with, instead of trying to worry about, like, what's the formalization of these models and does it have a name? So here's what I did. I designed something that I'm calling hybrid labs, which I'll explain in a second. And one of the features that I directly contribute or attribute to Wendell's inspiration is that these labs have discussions in them, which students are like, what? So they start out with these kind of theoretical discussions, just like the discussion labs that Wendell designed. And I want to, let's see, what am I doing here? Oh yeah, so I want to give you an example of that, because this is sort of a fantasy of mine to like get physicists to reason about biology. So here we go, you're going to have to do it. Okay, so maybe you knew this, maybe you didn't know this. Mutation rates are well, let me just say, mutation rates are the number of mutations per generation that an organism experiences, right? So from me to my child, there could be a lot of mutations across a generation, or there could be a little. So the question is, thinking about these different groups of organisms, which of these would you expect would have a higher mutation rate or a lower mutation rate? Or maybe you think all the mutation rates are essentially the same. Like, what would be your intuition about this? And why? And I would like you to actually think about this right now, just to do some biology for once. So go ahead and talk. Yes?
Is the rate like the number of mutations or percentage of genes that mutate?
It's the per base pair rate, if that helps.
Per base pair.
Per base pair. Per base pair. Does that make sense what a base pair is? Sorry. I forgot. You guys per base pair per generation. All of this is good thinking. It's fine. Just Okay, I'm gonna stop there, even though this is like so gratifying. Just hearing you all say like mutation and mechanism in the same sentence, even. Okay, I'm just curious if anyone's willing to share some of their thinking on this. Oh, right.
We were thinking B, because humans have more complex genetic material. Some of us thought in our group, although some of us also said maybe plants do. But then you said per base pair, so we
didn't get into that part.
Okay. What did you mean by complex? I'm just curious.
well, like more.
More.
Yes.
Cool.
Like a bacteria. Well,
That's
yeah,
good. That's good.
So larger
Right.
genomes are more things that could be made.
More.
Yeah, exactly right. So
More things, more
opportunities
genome.
for mistake
Yeah.
if you have more. But then you
Right,
can't
but then the
pull
per
those
base pair
out.
thing. Yeah.
normally.
Yeah.
So I'm trying to slot this into a thing that whatever the mutations are, that over time with natural selection effects, like there's got to be some sort of a success rate issue that's going to be playing a role here. And E. coli or the bacteria are living in a different environment and reproduce far more quickly than Homo sapiens or plants, if I'm thinking about long-lived plants.
Sure.
I don't know which plant you need here.
Even that plant. Yeah.
Much longer time than bacteria,
Right.
for example. And then my brain shuts off because I don't know, actually, I can't figure out how quickly a mutation affects survivability in the next generation, what effect that's going to have on the environment that you're in. And so I want bacteria to have the slowest rate, but you only have one option in there with bacteria, and that goes against my sense of how answers get questioned. So you don't have all the
Okay,
options.
it's a lot of
so-
thinking. But why did you want bacteria to be the slowest? Can you just say?
Because, or maybe as soon as you say back what I said, then I don't agree with myself. Because they're I feel like bacteria live in a more quickly changing environment. But actually, no, that was the debate. Like human beings on the time scale,
right.
from 20 years ago,
Right.
let's
I
call
see what
it
you're
generation
saying.
20 years.
Yeah.
Over 20 years, a lot of change is possible.
So they reproduce every 20 minutes. So right. This I wish I could keep going. This is the best, but but I can't because this isn't really the point of my talk. But you can see, you can see that it is possible to do reasoning about biology. It really just like that. And this is exactly what happens. All of a sudden, you get a room full of students talking about natural selection, talking about genome size, talking about mechanisms of mutation, talking about time scale, talking about selection pressure. All of the ideas from these models, really, that they've learned about in the curriculum, they're drawing them together and trying to explain this particular weird thing that we've posed for them.
At the end of this, my students would say, "What's the answer?"
I knew you were going to say that. You're going to have to buy me a drink. Okay, so then what we do is we... Then what we do is we give them some ways to continue to do work with this. Oh, yeah, okay. So we asked them to do some experimental design. So we switched the question and we focus now on like, well, when would you expect to get that a high mutator would have an advantage over a low mutator? And we do this then with a bacteria that have intrinsically different mutation rates. So then you can start to experiment and you can try to figure out when one or the other strain might grow more. And then we also include this computer simulation environment where you can mess with a bunch of different relevant parameters, like what kinds of mutations are we talking about, or what's the nature of the environment. And then we ask them to kind of bring all that together in a presentation, which again was kind of central to the discussion labs that Wendell designed. And gosh, I am going to go long. I'll try to speed it up. I want to show you a little bit about what happens next. They can design experiments. In this case, this group decided to design an experiment where they wanted to know if the high mutator would be able to digest lactose or mutate to digest lactose and grow more because it had this advantage of digesting lactose. And they found that it did, and actually this is sort of like a failure point in the design because it was like they knew that that was going to happen. And so there wasn't like a lot to think about. So you think about your intuition. If you put, this is like Michael's intuition, if you put an organism in a new environment, you want to be able to generate mutations. And if you can generate mutations, you're more likely to be able to adapt to that new environment. So students come in with that expectation, and then they confirm it with an experiment, and we're like, that's not what we want them to do. But then we design this computer simulation so that they can see that initial result. So here you've got two strains of bacteria. One is the high mutators in red and the low mutators in blue, and they can compete them, and they can see that initially the red high mutating strain in a new nutrient environment will have an advantage. But if you run the simulation for long enough, that'll switch, because eventually everybody can digest lactose, basically. And then once everybody can digest lactose, the only other thing that is going to happen to you is you're going to start accumulating bad mutations, right? So eventually, in the long run, having a high mutation rate might not be the best strategy. And students sort of just get shocked by this and try to figure it out then, why that could be happening. Why would a low mutator outcompete a high mutator? And then they start doing the thing again where they're reasoning, right? Then they start thinking about, well, what could be the possible mechanisms? And this is just some kind of jokey dialogue, but it gets much deeper. This particular student came up with some really interesting ideas in his lab report. One, and I just want to point out, one of these ideas kind of came from this simulation environment. Like, we had a lethal mutation as a parameter. And so that kind of, he's drawing on the resources, the model ideas that we gave him. But this idea about overshooting the carrying capacity and the high mutating strain then competing with itself and then crashing, like, that was his own idea that he just spontaneously came up with. My point really with that example is to say that if someone were to ask me like where the modeling is in these labs, it would not be correct to locate it over here in this computer simulation, right? Although, even though that's kind of initially how I thought about it. Like, we'll have an experiment and we'll have a model, and then there'll be some interaction. Really, there's this larger space of kind of ideas and models that students are drawing on, that this computer simulation represents some of those ideas that we want them to be thinking about, but some of those ideas they just come in with. And that's really where the reasoning is happening. This is just helping them expand the space of ideas that they're thinking with in this experimental system. How am I doing on time?
2 minutes.
Yes. All right.
Enough time for a short rant?
Sure. No, this is the uplifting portion of the talk, where I want to show you about how students experience this, encountering weird things and problems and unexpected situations in lab. And what we found in our interviews, and this is the same student who was like, what? In the computer simulation, is that they like it. Like, they're actually curious about what's going on in lab. He even goes so far as to say he was grateful that the first simulation showed this confusing result, because it allowed him to continue to think about what was going on and bring in these extra simulations and so on. And across all of our interviews, we have seen this pattern, which is that whenever students see something that goes against their predictions, where there's some mystery, where there's something that they do not know, where there's something that's the opposite of what they thought, they get excited and interested. And really, if I learned anything from Wendell, it was to cultivate this. This is what he was going for. He wanted students to have this sense of curiosity and wonder. And then the model ideas were just the things that were going to help them deal with that sense of curiosity and wonder and have some resources to begin to make sense of it. So ultimately, while Wendell was many things, it's this last thing that I like to think of him as. And I just want to share a really quick anecdote that Cassandra reminded me of, which is like just the image that I want to have of Wendell. This is not Wendell. This is a random child that I found on the internet in black and white to simulate Wendell as a child. But I like to imagine this is like Wendell as a child, sitting outside reading 20,000 Leagues Under the Sea, maybe, for example, and looking at the trees and noticing that there's wind in the trees, and starting to think about, like, what is wind anyway? Where does wind come from? And this is a story Wendell shared with us. And when he was a kid, he developed his own model of where wind came from. And it came from the trees, actually. The trees were pushing the air around, and they were the ones generating the wind. And he was watching these trees moving and thinking about his model of wind. And this is exactly the kind of thing, sorry, I wasn't really expecting it, there it came. This is the way I want to think about what Wendell is trying to cultivate and inspire. Making sense of the everyday world, having a sense of wonder, and then models are really just your way of putting together your ideas in order to do something with that. So, thank you.
Along with your changing the question of models about to models for, let me inject my engineering students' perspective, because to them a model, we're talking like models are there to build understanding, and that's us. But the engineering students, their model is there to guide them in designing something. So it's a complete reversal. I'm not using it to try to understand something. I'm trying to use it to guide me, because it's too expensive to crash cars until I see what's the best design. So friction is not something I take away to make my understanding better. It's something I put on to make it more realistic.
Yeah. I can say something.
Go ahead.
So I actually think, there's a lot that I didn't show about the way we use the computer simulations and the modeling ideas in the course that I'm running. But I think one of the ways to think about them is, not just explaining, but also sort of highlighting what you don't know. And you saw a little bit of that. And so that's kind of similar, I think, to maybe what you're talking about. So when students are messing around in the computer simulation, they can say, but we don't really know what these parameters are, like in real life. And so then they can say, oh, that means we need to go do an experiment. And so in that sense, the model isn't just explaining, it's showing a path forward to continue doing some kind of experimental inquiry.
I really liked this idea of students encountering something they don't know, and then being excited by that and the attempt to explain it. In physics context, what I see with students is when they encounter something that doesn't work, and we can problematize what it means to work, which is what I've been thinking about for like the last 10 minutes. Okay. They get really frustrated, and they don't know why it doesn't work or how to fix it. And then when they do, then they get really excited. And so I'm wondering if you can help me think through maybe like, do the students in your class go through this period of frustration before that period of wonder?
I think they do, yes. And there's so much more to the design of this course that I didn't say, but a large part of it is framing how you're supposed to be experiencing it, and also making sure that they understand that none of their grade is really related to them getting a particular result. So we do heavy amounts of framing. This requires a lot of TA training to say, like, the thing here is celebrate when their experiment doesn't work. Really cultivate a sense of, this is not the point of this. It's not that your data really are not that special or important. We just want you to be able to think about something. And so we have to hit that pretty hard. But I'm happy to talk more about it. Oh, is your time going?
One more question. One more?
I can talk.
Okay, great.
Thank you for that. It was really lovely, and and thanks for the teaching about biology. And so I'm wondering where your work with this development goes next, and if you have other colleagues in biology education who are partners with this. I think one of the things that this session is a testament to is the long time scale and the large number of people that were involved in the work that Wendell led and inspired. So, yeah, I mean, I guess where do you see that unfolding for you?
Is your question how am I going to influence the biology education research community, or what I'm going to do next?
Well, the second one, but also I hope you have colleagues and conspirators in there as well. Do you?
I do. I'm trying to figure out a good way to.
They didn't meet Wendell.
What?
Your
they did not meet Wendell, and I guess I'm a little bit weird in my community. I think that's not wrong to say that. But a lot of the biology education research community has come. It's an earlier it's a younger field. It just comes straight out of biology into education research. I had this experience with Wendell in the School of Education, a much longer trajectory. So I think I'm pushing out things that are not exactly happening as much in the biology education community, but starting to, for sure. So there are definitely some colleagues of mine that are doing similar things. But yeah, I'm in the early days, I would say. But, I'm a fighter, so.
All right. Should we thank Julia again?